Hi everyone, we are going to go over the first part of the gastrointestinal tract. So that's going to involve doing a little bit of review about the physiology and anatomy of the GI tract. So we're going to start out with that. Excuse me. So this is going to be a brief overview of motility and circulation so that we can learn the parts and then learn the function. So going on, what we will start with are the learning objectives, which are going to involve the general signaling within the GI tract. So these are excitable cells, so there are some action potential gradients that are generated here. There is circulation, there are contractions that respond to increases in circulation. And there are different types of potentials that are generated in the GI tract. Whenever I say the term tonic, that refers to tone. Like there's a baseline control with a lot of different objects, which we revolve to or refer to as our tone, just like muscle has tone. So when I say tonic, that has to do with general tone. And then there's more phasic, which is more integrated types of action potentials that are going to result in some sort of large amount of activity. Okay, so just going on, we will then talk about the gut-brain system known as the enteric nervous system and how that controls motility. Because this involves a lot of smooth muscle, we'll talk about reflexes, and a lot of the reflexes here are pretty straightforward. Remember from Physiology 1, you may have talked about, I know I talked about it with you, the myotonic reflexes, and that has to do with the fact that when smooth muscle is stretched, it responds by contracting. What that means is in a large part of the gastrointestinal tract, pretty much all of it, we're going to have increased motility, increased action potential propagation when there is the presence of a substance in an area. So stretch will respond in contraction. And some of these are involved in the reflex arcs that we'll look at. We're briefly going to go over or at least present some hormones that you will have to know for this chapter and the subsequent GI information. There is a full secretion chapter though where we go into the very deep info involving all of these different secretory products, and you do have to know all of them for your boards. And then finally, we will touch upon the blood flow in this system. It is the largest amount of cardiac output in normal parasympathetic circumstances. That's why we refer to it as the rest and digest system. About 40% of cardiac output is going to the splenic circulation, which involves the majority of the lower GI tract. Okay, so moving on, we are just at general GI physiology, and in this picture we have the main muscles and associated organs involved with movement through the gastrointestinal tract. So we will start up in the mouth, but the mouth and swallowing will eventually lead to the esophagus. And what I want you to notice here in this picture is when we have just a normal anatomical understanding of where the esophagus enters the stomach, it's a weird type of angle that's created. So the fundus, or the top of the stomach, is located up here, right underneath the pectoral muscle in this person. We have a sphincter right in this area, which helps keep the stomach closed. But I want you to notice that if the esophagus was truly just up at the fundus of the stomach, then we would really never have any issues associated with heartburn or GERD. So it's really a lot of just anatomic variation that leads to that being a common disorder. And then the stomach, we know, is our organ of mixing. That's where food goes in and mixes with acid in a phase called chemical digestion that breaks these macromolecular structures down into usable parts. And at the end of the stomach is the pylorus, which helps keep the stomach shut while the stomach is mixing these contents. And when it's time for that food to move on, the pylorus is going to let out small amounts of food at a time into the small intestine, beginning with the duodenum that we see connected to the stomach. And then the small intestines involves the other two parts that we'll talk about, the ileum, which then leads into the large intestine, which we can see has these large flexures there. And just in general, know that the small intestine is about 10 to 11 feet of coil, whereas the colon is maybe 2 to 3 feet. So it's a pretty large size difference. And then eventually the large intestine leads to the rectum, which will allow the excretion of food out of the body. So there's two terms that I said on this slide that I want you to take note of, and on the slide prior. When I said secretion, so when things are secreted, that typically means into the body, more specifically into the bloodstream. When things are excreted, it means that they're removed somewhere outside of the body. So one fun fact about the gastrointestinal, or if we go to the next slide, the alimentary canal, is that everything in this long lumen from the mouth down to the rectum is technically external to the body. So that's something interesting about it. And that makes sense if you think of the stomach forming an ulcer and then bursting and dumping its contents out. That causes something called peritonitis, or inflammation of the peritoneum, the lining of these cavities. If the appendix bursts and you get appendicitis, that is very damaging and can lead to peritonitis and something called a fistula that I'll explain to you. Same thing with the colon. If the colon perforates and empties out its contents, we get, again, peritonitis because this mixing of fecal material into the body cavities. So we want all of those things inside of the alimentary canal to stay there. And if peritonitis doesn't heal on its own, you get a lot of scar tissue involved because of the type of inflammation. We call it fibrinous inflammation when an organ is damaged and necrosis, and we get some healing processes that come and complicate things. And those increased amounts of scar tissue can lead to what we call contractures in the organ itself, but adhesions where scar tissue connects pieces of the GI tract or other organs either to each other or to the lining of the cavity, to the wall of the GI tract, to the abdominal cavity. So they're very bad types of situations, even when they do heal properly. If they don't heal properly and those bacteria gain entry to the bloodstream, that is called septicemia. And you've probably heard that referred to as a patient going septic. That means that bacteria have now gained entry to the bloodstream, and that's a life-threatening condition. So even though it's a lumen that runs through the innermost cavity of the body, right, in a pretty long straight line relatively, it's still considered external. So when we say excretion, that can involve the squirting of things essentially into the GI tract. So if the liver produces bile and that's moved to the small intestine, then that's an excretory process. When fecal material is removed from the rectum in digestion, in going to the bathroom, essentially, that's excretion as well. So the main processes and functions of the GI, or alimentary canal, are to provide the body with the correct amount of water, and we'll learn all about absorption. And just before we go into anything else, knowing that the majority of absorption happens in the small intestine, primarily the duodenum, that's pretty important. So we're going to absorb pretty much everything that's important in this first area, in the duodenal area. It's the smallest area of the small intestine, but it's also the busiest. This is also where we absorb nutrients, electrolytes, drugs, therapeutic substances, everything generally, besides two things in the world, they're absorbed in the small intestine, primarily the duodenum. And if we go back and think about diffusion that we learned about in the very first part of Physiology 1, things diffuse from high to low concentration. So water and nutrients and electrolytes, they'll be high in concentration in the duodenum, but low in concentration in the external extracellular environment. So that's a pretty passive process. The other things that it does is not only absorbing these things, but it has to prepare them for absorption. It has to make them water soluble, and it has to move things to the areas that can best utilize these substances and move them to the areas again that need them. So we have movement, we have different secretory products, we have digestive products, there's absorption, we have to have a readily available blood supply in these different organs so that they can receive the products of digestion. They go into the bloodstream. And most of this is just under the control of local systems, the gut-brain system or the enteric nervous system, and then with the overriding ability of the parasympathetic or sympathetic systems to increase or decrease the general tonic activity. So on slide 5, the alimentary canal is breaking down what we call the major organs of digestion. We also have accessory organs that help out. So if you think about the mouth, for example, that's definitely helpful to have. The teeth are helpful to have, but they're accessory organs in that if you didn't have them, that doesn't mean that you won't digest properly. So how we define digestion is breaking down food into the molecular components for absorption. All of the food that we take in, they're in the form of macromolecules, and those macromolecules have to be broken down into more molecular substances that are water soluble so that they can be absorbed and utilized. So we'll go through the different areas, and I want you to know already that there are 2 different types of digestion. There is chemical digestion, which involves some sort of secreted product, whether it be an enzyme, whether it be water or acid. Those are forms of chemical digestion that breaks down the bonds of these macromolecular structures so that we can liberate the useful parts and separate them from the parts to be excreted. And there's also chemical digestion, which has to do with some sort of muscular breakdown of food. So if we go through these major organs of digestion, we begin in the mouth, and the muscular type of digestion there, mechanical digestion, is chewing, or what we call mastication. The primary component that's made in the mouth for chemical digestion is saliva. So saliva is a huge amount of water. It is a very stable pH. It involves a very tight regulation of the transit of chloride and potassium and bicarbonate with sodium to make sure it has this nice basic type of constituency. And there also is an enzyme in the mouth. There's 2 of them. One is in the saliva, and it's salivary amylase. And you might remember that if it ends in ACE, it's an enzyme. So amylase is an enzyme that starts to break down starches. So that is anything sugary, any kind of carbohydrate. That breakdown begins immediately in the mouth. The other product is secreted by the tongue. It's called lingual lipase. Lingual lipase is secreted not from saliva, but from cells in the tongue that help to break down fat. And that really influences not only the solubility of things containing fat, but also the taste of that food. Humans enjoy food with a lot of mouthfeel, a lot of that silky feeling, and lipase helps break that down so that it can be swallowed. Moving on to the esophagus, I want you to know that the esophagus has no interaction with chemical digestion. There's plenty of cells lining the esophagus that do secrete things, but primarily it's mucus. And that mucus is there for lubrication because the muscular action of peristalsis by the esophagus, its job is to create waves of contraction that move food from the mouth, where it's chewed, into what we call a bolus, and then that bolus has to move down the esophagus to get to the stomach. And that peristalsis only moves in one way. There's no such thing as reverse peristalsis. We'll talk about vomiting later, but it's not just reverse peristalsis. There's something called the law of the gut, which states that everything is polarized towards the anus. So that means we're not going to have any reverse directionality of contraction. So from the top of the esophagus, right underneath the pharynx, that's where the esophagus starts with these waves of peristalsis, which move food down this bolus down to the stomach. So nothing chemical, there's only mucus there for lubrication, and most of it is muscular. So that's its peristaltic movement. And that is mainly to move things. So that's a propulsive type of movement. Then once the sphincter at the bottom of the esophagus opens and allows entry of that food into the stomach, there is a huge amount of chemical digestion and just as much mechanical digestion. There's a large amount of acid secreted by the stomach. So the acid secreted by the stomach, again, we have a whole chapter on this, but there's two of them. There's hydrochloric acid, and then there is pepsin. So I want you to remember that the P in pepsin stands for proteins. And you can also help yourself remembering that by thinking of the term peptic ulcers. Peptic ulcers are ulcers or sores that form in the stomach due to something breaking down the mucosal barrier. So that means you're having destruction of the stomach by the enzyme there, or this acid there, pepsin, which breaks down proteins. And remember, all of our organs are proteins. So pepsin is something that's good at breaking down animal cell walls. So things that are dairy, are meat, eggs, milk, all of those things require pepsin for digestion. Hydrochloric acid can break down anything, but it's primarily good at breaking down cell wall components so that you can break down your celery, your salad, things of that nature. Another way to remember this too, we'll talk about drugs that people take for GERD or take for different bowel disorders. They're called proton pump inhibitors, and they only affect hydrochloric acid secretion. And if you think of patients with Crohn's disease or ulcerative colitis, you typically don't have them eat raw salads, raw vegetables. They can actually end up in the emergency room for that. What you'll see is they're on these medications to inhibit hydrochloric acid secretion so they don't get good breakdown of plant cell walls. But also that amount of roughage means that you're going to have a lot of contraction in both the small and large intestinal muscles, which is good if you have a healthy bowel, but it is not good if there's a diseased bowel. So we've got our peptic acid and hydrochloric acid. There's also a huge amount of gastric juices that are made. Those are going to include water, include mucus, include these acids, and that's going to make up our chemical digestion. We're going to make a really low pH environment for all of these things to be broken down into component micromolecular structures. So that's your chemical digestion. And then we have the muscular digestion in the stomach, which is called churning. We'll see that the entire gastric wall is made up of rugae, which allow the stomach to expand. And then underneath the rugae in the submucosal barrier, we have a gastric muscle. And the stomach churns by making these nice coordinated churning movements to break down the food. So remember, we had a bolus when it was swallowed, and the stomach makes it into something called chyme. And chyme really resembles something like a cake batter or even a paste. And only when it's in the consistency of this paste is it ready to move on to the small intestine. So we'll talk about when there's alterations to the acid secretion or the churning capabilities of the stomach. What you'll see is instead of a paste-like substance entering the small intestine, we have pretty undigested food in some cases. That's called dumping disorder. We'll talk about it in just a second. But you would see undigested food in the stool from that patient. So we'll get there in just a little bit, but our stomach, we have chemical and mechanical digestion in churning and the acids. Something that's technically accessory, but we definitely need it considering the bulk of our diet is going to involve phospholipids. It's the liver. And the liver, in role with digestion, is going to make bile. That's the most important thing with regards to digestion coming from the liver. There's no pancreas in this picture, but we'll see that the pancreas is also very important because it makes bicarbonate. And bicarbonate is a highly basic substance, and it's going to be excreted into the small intestine so that the low pH acid doesn't damage the small intestine. And bicarbonate and products from the pancreas are released at the same time as bile is released into the small intestine through this nice anatomical process. So the liver, there's no mechanical digestion involved. There's just the flow of bile in the very small places called the canaliculi. It's truly just passive. These cells make bile and they flow down their channels and they fill up this common bile duct. And when there is the presence of food in the duodenum, it signals to the sphincter of Oddi, so this hepatopancreatic sphincter, so that we get contraction of the gallbladder and then a forced opening of the sphincter so that those things are emptied out there. So it's involved in chemical digestion, but the liver itself doesn't do any chemical digestion with regards to the alimentary canal. It is going to deliver bile. Okay, so it's technically accessory. There's nothing motor here. We're just moving smooth muscle contractions to bring the bile down. So we get the emulsification or the breaking down of fat so that we can absorb fat products and then also excrete different products that are fat soluble in the feces. Without bile, that doesn't happen. In the small intestine, there is a lot of secretion, but it is mainly mucus, again, and some water to help with the movement of food and aid in the absorption of the molecular substances that we would like. The small intestine does some peristalsis. It's a little bit different. It is going to be the propulsatory movement that peristalsis occurs in one direction, but there's an additional type of churning or mixing movement that occurs in the small intestine called segmentation that we'll take a look at the end. And everything stays in the small intestine. It's actually a long time. It can be up to 10 hours in some individuals until everything good is sucked up out of the meal. Then the ileocecal valve, so that'll help you remember the ileum is the last part of the small intestine, and it's attached to the cecum, which is the first part of the large intestine. The ileocecal valve opens, and what enters the large intestine is a mix of waste food and cellular debris, cells that have sloughed off the wall. And then the large intestine is there to move things towards the rectum so that they can be prepared for expulsion. The main goal of the large intestine is to absorb water. Remember I said, though, the main part of the body that we do absorption with is the small intestine. So just because the large intestine only absorbs water doesn't mean it's the main organ that does that. The small intestine is still where most of the water gets absorbed. However, the large intestine will suck up everything that it can that's left, and you can tell how long things have been in the large intestine by how firm the feces are. So if they're watery feces, it means that it made a quick transit through the large intestine. That's called peristaltic rush. We'll talk about that later. That's a fun disorder, or it's really just diarrhea. But the longer that it's been in the large intestine, the more firm and difficult to pass the feces are, and the more malodorous they are. So those are the different major organs associated with the alimentary canal, and they are considered the major organs of digestion. Again, liver's technically accessory, but we do need it in order to digest. All right, moving on. Some other things anatomically that are important are the sphincters of the stomach. We'll get to swallowing in a little bit, so just know that there is a small sphincter up at the top that is called the upper esophageal sphincter. So the upper esophageal sphincter is up here. So we call that upper is with a U. So I'm picking a color and talking at the same time, and that's not going very well. All right, so we have the upper esophageal sphincter up here, and then we have the lower esophageal sphincter down here. So the upper esophageal sphincter should be closed most of the time. The only time that we should have opening, of the upper esophageal sphincter is when we physically, oh man, this drives me nuts, swallow. So when you push your tongue up towards the roof of your mouth to physically swallow fluid or a solid substance, that forces the upper esophageal sphincter open so that the food can then, or the bolus can enter the esophagus, and then it'll be trafficked down until it gets to the lower esophagus. So the lower esophageal sphincter opens when food stretches the area right above it. And the lower esophageal sphincter should open briefly just to allow transit of the bolus, and then it closes very tightly afterwards. Secondly, we have a pyloric sphincter, and the pylorus is a little bit different because the lower esophageal and upper esophageal sphincters are always closed unless there's physical swallowing, which will open this one and then subsequently open the lower one once the bolus reaches there. When there's no food in the stomach, the pylorus valve is slightly open. And why that is important is because we have the free transit of fluids then through the pyloric valve. So that means when you wake up in the morning and there's nothing in your stomach, but you have a nice cup of coffee, the first sip of coffee is going to make it down to the stomach. It doesn't really need to hang out here because there's not a ton of good things to absorb from it. Pretty frequently, it just goes right through this little open hole of the pylorus into the small intestine so that we can absorb the water from the coffee, then also the caffeine. So it seems like caffeine on an empty stomach hits your brain and makes you feel awake a little bit faster. And that is anatomically true because there is an opening of that pyloric sphincter when the stomach is open, or not filled. When there is food or liquid in the stomach, the pylorus is very, very tightly shut. And that's to prevent anything that's undigested or not in a liquid formation to just travel on into the duodenum. It also is important for us to maintain our pH balance because if you have some like lemon water in the morning or some vinegar or something that is really off with the amount of pH, or even if you drank some old school sodium bicarbonate in water, if you have heartburn or a stomach ache, that'll make you feel better. But if it goes to the duodenum too quickly, it'll raise the pH of the bloodstream and then the body. And then you have something called alkalosis. So the pylorus controls how much of something can move on to the duodenum so that we don't have any large alterations to electrolyte balance, pH, even dehydration type of balances. So just for some simple disease purposes, when the lower esophageal sphincter fails to close, which could be because of stress on the sphincter, it can be from the stomach being too full, it can be from laying down on your back after you've just eaten. All those things lower the tension here and make it harder to stay shut. So if this fails to close completely, you now have this very low pH stomach juice, essentially, coming on up into the esophagus. And the typical pH of the stomach is 2. So anywhere from 1.5 to 2 is a normal stomach pH. So that's going to hurt quite a bit, and that's called heartburn. You guys know that. That's a pretty painful and also common thing. 1 out of 5 Americans report having that type of heartburn. So failure of the lower esophageal sphincter, well, that's an interesting shape, to close is GERD. If we have a failure of the pylorus to close correctly during churning, that is called dumping disorder, which is pretty much just what it sounds like. It's important for food to stay in the stomach for it to be turned into chyme. That's usually 1 to 3 hours. So 1 to 3 hours. Why do I keep pressing that button? 1 to 3 hours. And the thing that helps it stay in that area for that long is the protein or fat content. So if you eat something very low in protein or fat, you'll have quick transit of that food out through the pylorus into the duodenum because it doesn't take that long to break down. That also means that you don't feel full that long because now your stomach's empty an hour later. For something really high in protein and fat, we have to keep the pylorus shut much longer, and it takes about 3 hours to break down a full high protein, high fat meal. If that food is not there, that 1 to 3 hour ratio, you'll have undigested food empty into the duodenum. So that is very painful. So imagine having some tempura veggies, which are delicious, a nice little tempura piece of broccoli you just ate, but you have dumping disorder. What that means is it's going to hurt as it transits all the way throughout your small intestine and then large intestine. And then what you'll see is that patients will have undigested food in their feces. So it's important to get that investigated. It can be treated. There are some different surgical and pharmacological ways to address this. But dumping disorder, not super common, but what is super common is heartburn or reflux, and that's just caused by improper closure of the lower esophageal sphincter. Okay, so sphincters are important. Other things that are important that you've probably gotten a little bit in other courses, but if not, let's just go through the layers of the GI tract. So everything in the abdominal cavity. So let me just back up for a second. Here's our abdominal cavity, and then we have our thoracic cavity. Remember, the diaphragm is what separates the thoracic from the abdominal cavity. So most of what we're talking about in the GI tract is below the diaphragm, right? Because it would be right here. The stomach should be underneath it. The liver should be underneath it. The only part that we have in the thoracic cavity then is really the esophagus. So that's going to be the only thing that will really differ in these walls. Because if we have something in the thoracic cavity that has an adventitious lining, meaning it's more tough and more similar to scar tissue. If that organ lies in the abdominal cavity, it's going to have a serosal membrane, which are very thin membranes, making it easy for things to transit between the different areas. So for everything besides the esophagus, this will hold true. The only difference, again, is instead of serosa here, we will have adventitia. Okay, so first, the outermost layer is the serosa, so nice, soft outermost layer, kind of protecting everything from the external environment, but also allowing a nice watery surface to encase the GI tract, which is designed to work underwater. Then we have two different types of muscle. We have longitudinal muscle, which is named such because it goes straight down or in a line. If we think about a big line of the small intestine, longitudinal goes the whole length of that piece of esophagus or small intestine or large intestine. Then we have circular muscle. And circular muscle is different than longitudinal muscle, which just acts as a sheet and it all contracts together. Circular muscle is like a series of bands. And just like rubber bands or hair ties, they contract circularly. So when they contract, they make this hole in the middle smaller, and they contract kind of one at a time or sequentially. So 1, 2, 3, 4, 5. So longitudinal muscle helps to move things through in one nice long contraction, and it has to work in a perfectly orchestrated way with circular smooth muscle so that things can be moved from point A to point B. So serosa, longitudinal, and then circular muscle. So longitudinal is going in one direction down the whole length of the organ in a straight line to make a sheet. The circular muscle is a small band that encases each segment of this tissue. Underneath the circular is the submucosa, literally meaning under the mucosa. And the submucosa is where all of the fun extra stuff is, like the blood vessels and the glands that secrete things. There's also lymphatic vessels here because the holes in lymphatic vessels are much bigger, making it easier for proteins and fat to go into there first. Then you have the mucosal layer, and depending upon which area of the GI tract you're on, it's either really, really thick or relatively thick, but the mucosal barrier is very strong, and it makes the nice mucosa, or just mucus essentially, thick mucus that is highly basic, which helps lubricate, but more importantly, helps to protect the organ itself, because we have all of these low pH contents that we don't want burning a hole through any piece of this. And you might know that that does happen when there is damage to the mucosal barrier, or that's what causes ulcers. So that can happen because of a bacterium called H. pylori, and then the two things that you take in that cause the most amount of ulcers are non-steroidal anti-inflammatory drugs, primarily aspirin. And then the other thing with aspirin is alcohol. Those are two very, very damaging substances. Then we have the first little mention of the different plexi. So we have the enteric nerve plexus, Meissner's nerve plexus. We'll talk about what those do and what they control, but they're pretty straightforward, I promise you. They won't be confusing. This is an actual micrograph structural tissue stain of what the esophagus looks like. So again, as I mentioned before, because this is in the thoracic cavity, notice that we do have an adventitious lining here. So here's the adventitia, this blurry white part. So that's made out of different fibrous structures. In the same way that we have nice strong scar tissue, the adventitious lining helps to protect the esophagus, and it means when you pick it up, it's not squishy. If you've ever watched a video of the small intestines being taken out, they're very rubbery, they're very squishy, they slip through your fingers. The esophagus isn't like that. It's pretty tough and it's hard, and it doesn't move a lot like the small intestine does. So here we have all these pieces. We have the mucosal piece. So that is made of stratified squamous epithelium. What that means, if you're interested in cancer, is because it's squamous tissue, then you can have squamous sarcomas there, squamous carcinomas. So when we talk about like esophageal cancer, it would be generally a squamous cell type of cancer in the esophagus. We've got the submucosa area, again, with all of the connective tissue, but also the blood vessels, the lymphatic channels, and the secretory cells that make their products. There's the muscular level. We have our circular and longitudinal muscle. Remember, longitudinal is in a nice long sheet down the whole length. So looking at this guy, the longitudinal muscle would start at the top of the esophagus and go all the way down. The circular muscle, there'd be a band here, then here, then here, then here. It'd be like wrapping rubber bands sequentially all the way down the length of the esophagus. And it's important that they work together. If they don't, it causes pain. That is the main reason why there's pain with intense diarrhea or cramping. It feels like searing pains across the abdomen. That means that there's a mismatch in communication between the circular and longitudinal muscle. Things are just moving too quickly. Okay, so as I mentioned, especially the longitudinal muscle, we've said this word in Advanced Physiology 1, and we say it in Pathophysiology when you take it 2. The word for tissue that works together as a sheet, because all of the cells are connected via gap junction, meaning you need one cell to be excited for the entire sheet to then sequentially be excited. So very similar, if you want to picture this little nucleus as a guy at a football game. If he starts the wave, that wave will spread to all of the other pieces in a sequential fashion because they are all attached. So these tissues act as a sheet. So GI muscle is organized this way, nice parallel fibers. And remember what gap junctions mean is we don't need to release something over a synaptic cleft for it to bind to receptors across the synapse. Instead, we have an open door between cells to allow the free movement of ions from one cell to another. So the stimulation of one of these cells will spread across so that the tissue acts as a sheet. So we call that syncytial muscle, and a singular sheet is a syncytium. So smooth muscle works as a syncytial tissue. That smooth muscle in the area, as I mentioned before, we have the normal tonic activity, so the normal tone of the GI tract. So if you are at your computer right now and you just want to shake out your right arm, and as you're shaking it, put your fingers on your bicep, you'll notice that your bicep's not just flopping around. It doesn't become completely floppy. It doesn't lose all of its tone. If you did have a nerve lesion there and you lost all innervation to that muscle, then eventually you would have a floppy muscle tone. But all muscles, skeletal and smooth muscle and cardiac muscle, they have a basic level of tone. So they're in a semi-contracted state all of the time. So we call that, in neuroscience, we call that tonic, because it makes sense, right? That's your normal tone. Sometimes you'll hear this called vagal tone because the GI tract has such an important integration with the parasympathetic nervous system, that the parasympathetic nervous system is going to have this general tone being established. And if we measure from a cell in the GI tract, when it's just resting, hanging out, you're not doing anything special, it's just doing its rest and digest functions, it's just going about its normal resting activity. So that's similar to what would happen if I put an EEG on you over your brain and you're not doing anything. We actually call that default mode, when you just kind of space out for a little bit and then you come to. That's your brain entering default mode. And that's what these waves look like. Your brain's just going about its business and it's engaging in its normal tone. So the slow waves, when we say rest and digest, just normal rhythmic gastric contractions, it takes about 0 to 4, it's not 0, it takes about 24 to 48 hours from things to make it from your mouth to the toilet. We call that mouth to anus time. It's a great term, or gastric transit time is the other term that's more physiological, but it's the time from eating to defecation. So 48 hours typically, that's a long amount of time. That means when you eat a taco for dinner, it's going to eventually be excreted. If today's Monday, it'll be excreted on Wednesday. So that's going to go through a nice long transit through your body. And as those things are moving, it's just going to involve normal tonic, slow contractions, moving things from one place to another. So these aren't really action potentials if they do sum up. Remember, we learned about summation. So if you have a lot of ion entry at one time, you can get an action potential. But generally, slow waves, they just indicate resting. Now take a look here. So when there is summation in such a way that we do have action potentials that are initiated, we call those spike potentials. So that involves a true action potential being created, and it means something is happening and making something contract more strongly than it typically would. Primarily, this has to do with stretch. So let's pretend we're measuring from the stomach or to the esophagus. Let's just do that. We've been up there. So typically your esophagus is just going to be hanging out, doing its little tone, but now you took a bite of that delicious taco and it's stretching the area of your esophagus. So all of that stretch is going to excite these cells. We're going to have a bunch of cells in that area involved in that stretch response, meaning that positive ions will flow in and then spread down that like a sheet. So that's a spike of high activity. And then if you remember, sometimes when you swallow something, it doesn't go all the way down at first, or you have to work really hard to make it go down because there's not enough liquid or it's dry. So you'll have spikes which engage your muscles to contract, again, in a more vigorous way than they typically would. And then it returns very quickly back to slow wave. When there's a high amount of activity, so if this was the stomach and it was doing slow wave while you were eating that taco, and then the taco is now in your stomach, you're going to see a high amount of spike potential because now the stomach has to turn. Okay, so we believe that these cells of Cajal, we call them pacemaker cells, and I'll explain what that means if we haven't already gone over it. But pacemaker cells, just in general sense, before I get into the more complicated physiology, are cells that generate their own potential. So if you think about a pacemaker being inserted into the heart, it makes that heart have a normal rhythm. So it starts or initiates action potentials or rhythm. So your GI tract has its own cells like that. The cells of Cajal, they're pacemaker cells, and it's their job to help spread that polarization so that things move. All right, other things that generate spikes. Acetylcholine. Because the parasympathetic nervous system is the rest and digest pathway or nervous system, acetylcholine is released from the nerve, specifically the vagus nerve. So when acetylcholine is released, we have more parasympathetic activity, which is always going to be turning on the GI tract. Parasympathetic activity increases GI activity, which means number 3, parasympathetics, those are drugs that mimic normal acetylcholine. So when you give a patient cholinergic drugs, there's an increase in GI activity because it's mimicking acetylcholine, which is naturally released from parasympathetic nerves. Okay, so almost everything in the GI tract, again, it's stimulated to contract by stretch. Number 2, that acetylcholine, that's normal activity. Whenever acetylcholine is increased through parasympathetic upregulation, through you doing that, there's a way to do it, like the bear down response, like when you go on the toilet, hopefully, or wherever else you want to bear down. But when you bear down to have a bowel movement, you force the body into parasympathetic stimulation. And then giving a patient a cholinergic drug, that all increases GI potential and spike potential. When you give something that is sympathetic in nature, that hyperpolarizes it. So we should know that by now. The parasympathetic nervous system is going to turn on or generally depolarize activity of the GI tract, where sympathetic activation will hyperpolarize GI tract activation. Remember, this is smooth muscle. So when we learned about smooth muscle in Phys 1, you learned about the calmodulin mechanism and how calcium coming in combines with calmodulin to contract in a very slow and long-lasting manner smooth muscle. So slow opening channels, long duration action potentials, and those are what normal baseline activity of smooth muscle looks like. Slow waves, which are generated by these pacemaker cells, and spikes, which happen with stretch, acetylcholine from the vagus, or parasympathetic drug activation. Okay, I'm just taking a little look here, and then we'll go on. All right. So as I mentioned, the GI tract has its own nervous system, sometimes called the gut-brain system. And enteric, if you've ever heard the term enteritis, enteric means small intestine, essentially. But in this case, it traverses the entire gastrointestinal tract. So the enteric nervous system is a constantly running native nervous system that keeps your GI tract running without any input from the rest of the body unless necessary. All right, so we mentioned that we have sympathetic override. In this picture, those cells are red. We have parasympathetic override. Those cells are red also. But I want you to notice that those really just overlay these other natural plexi or nervous systems. So here is gastric epithelium. We can see that we have some cilia there. And I mentioned enteric. and small intestines. When we go further through these chapters, you will learn, and hopefully learn forever, that the small intestine has these villi on it. So that's how we can tell in this picture we're in the small intestines. So these villi are only found there, nowhere else in the body. Okay, so there are a couple of things going on here that are sending messages to the paravertebral ganglia, which are the ones near the vertebra, the sympathetic ones, to the cord or to the brainstem. So those can be things that get information from sensory neurons. There's sensory neurons, and in this case, we keep saying stretch. They are there to sense stretch of the tissue. So sensory neurons, they can send that information up to the brain, up to the cord to engage in local reflexes if necessary. But they provide the main source of information to these two different plexi. The myenteric, which is deep lower, it's down lower, so think about the layers. We've got serosa, we have our different muscular layers, longitudinal and circular, and then we have the submucosa, which contains the fun stuff. Remember, that's where the blood vessels are, everything else, the glands, but it also has these nervous areas. So we've got the myenteric plexus. Remember, myo means muscle, and submucosa means underneath the mucosa. And plexi means web. It's a complicated web of nerves. So knowing that, moving on, the enteric nervous system, it lies in the gut wall. It goes all the way from the esophagus to the anus, and it controls movements of the GI tract and the secretory products. It does its own thing. However, it can be altered by the sympathetic and parasympathetic nervous system. So if you had enough of listening to me and you need to go stretch or go for a run or go take out a load of laundry and you run down and then back up the stairs and your heart rate goes up, that means you're now engaging in sympathetic outflow. So the sympathetic nervous system will be activated so that it will shift the blood flow from your GI tract to places like your skeletal muscle and your heart in places that need it. So the sympathetic nervous system can come in here and shut off both of these things, because whatever your GI tract was doing before you engaged in exercise is not important anymore. The sympathetic nervous system will turn it off. And then you go and sit back down, turn on your computer again, your heart rate goes down, which means your parasympathetic will then need to turn on. So it will turn on and it will give the go signal back to both of these plexi again. So that will turn it back on, where sympathetic turns it back off. And here's a little breakdown, but their names tell you what they do, and there's a slide for each of them. The myenteric plexus, because myo means muscle, that is mainly and almost entirely in control of the gastrointestinal movements. So the movements of propulsion and the movements of mixing. Whenever we need stimulation of the circular longitudinal muscle, the myenteric plexus are nerves that signal to those muscles that tell them to contract. The submucosa, remember what's there. There's blood vessels and there's secretory cells. So when those plexi activate their surrounding environment, they activate the secretory cells to make and release their products. And they also will affect the blood flow, which might need to increase in that area if there's lots of activity, or it might need to decrease if that activity has ended. So the next two slides are saying just what I mentioned. So the myenteric plexus, nice long linear chain, goes all the way down the GI tract. It lies between the longitudinal and circular muscle because it needs to signal to both layers. So it's like the cream in an Oreo with those two muscle layers. Whenever the myenteric plexus is stimulated, the increased tone of the gut wall is achieved. So instead of being just relaxing, it contracts more easily because the tone is better. It increases the intensity of rhythmic contractions so that when there's contraction, it's not just slow and there's lots of space in between them. They're stronger and they're harder and they're faster. the rate and rhythm of the contractions goes up, and it increases the velocity of these waves of excitation. So if everything works harder and faster. There are some inhibitory neurons associated with the myenteric plexus, and its main goal there is to relax sphincters. So if we contracted a sphincter, remember sphincters contract in a circular way. So if you put your fingers like the OK symbol, and then you contracted your fingers so that your pointer finger is kind of burrowed into your thumb, so it looks like a snail instead of the OK symbol, that would be what a sphincter looks like when it's closed. So obviously, if we're contracting muscle to move things around, we don't want to contract the sphincters, or else we won't have any free movement. So there's inhibitory neurons associated with the sphincters so that they can relax and allow transit of things from one place to another. Finally, we have the submucosal plexus, which lies in the submucosal area. It is specific to each system because there's different products in each system. Excuse me, so we'll learn about that in the secretion chapter. So it controls all the local absorption, secretion, contraction of submucosal muscle. Okay, so for the first part, I'm going to stop there, and then I will pick up with the, well, let me go here because this is a good way to end, and then I'll move on to the more specific types of nerves. So just in general, I'll pick up here again, but remember, sympathetic is going to be inhibitory with regards to the GI tract, and the parasympathetic is going to be excitatory with the GI tract.